I think a long time ago, we got really interested in this idea about cancer cells have to interact with other cells, right? That cancer cells can't do what they want or do what they cause sickness by acting on their own. And so this ability to try and understand this, what I call cancer in context, meant that we had a real problem because how do we understand this in context, right? Because it's such a complicated landscape to try and understand this. So what I thought I would do is just share a couple of slides just to give you some context for how we think about the problem, and then go into a tiny bit of detail about what it is we're doing. So hopefully you'll be able to see my slides. And I'm not. Oh, it says it's disabled screen sharing. Can somebody activate that? Oh, still says it's disabled.
All right, bear with me for just a second, Dr. White. Let's see if I can allow the permission. We did have to get flexible just now when we decided to move to slides. Let's see. Share screen, advanced sharing options. Okay, this should work.
Okay, that works. And like I said, I'm going to keep this. Hopefully you guys can see this.
I can.
Okay. So I'm going to try and keep this pretty brief. But basically, if I were going to summarize the way my lab thinks about cancer and how we think about understanding cancer and ultimately treating cancer, it could be described as understanding cancer from the inside and the outside, right? And I think that kind of forms the nucleus of how we think about the problem. What do I mean by that? Well, what I mean by that is that all cancers, and I happen to study melanoma, but it actually doesn't really matter what you study, they depend on things, factors that happen inside the cancer cell, like mutations, like the gene BRAF, really common mutation in melanoma. But equally, all cancers then depend on its neighbors, right? And melanoma, for example, is surrounded by lots of other cells in the body, like adipocytes and fibroblasts and lymphocytes or immune cells, nerves and macrophages, and it's vast, right? The number of things that each cancer cell is ultimately surrounded by sort of dictates whether the cancer is able to proliferate, migrate, metastasize, and all that. And I think one really potent way of looking at it is actually, this is a slide of a patient at Sloan Kettering. There's a patient with a very advanced melanoma. And I think in this one slide, I can actually encapsulate just how complicated this inside-outside problem is, right? So this was a patient who had a biopsy, who had a lesion on their skin. And if you look at this slide, the purple cells here, and I'm just going to pull out a laser pointer here for a second. The purple cells here, which are kind of all these cells here, are the melanoma cells. But everything else in this biopsy is all non-melanoma cells. It's all the other stuff that surrounds the melanoma cell. And I saw this slide early in my career, and I was completely struck by this, right? Because it tells us that while much of our energy in the cancer biology field has focused on these cells, if you just look at this one slide, those other cells, the surrounding, the so-called outside, actually are by far, by number, the dominant thing. And so we have this hypothesis, actually, that it's this interaction between the inside part, the melanoma cells, and the outside part, all the other stuff, that actually determines how cancer cells act. And maybe we could use that for therapeutic, advantage. There are lots and lots of interesting cells that are outside. So these early melanomas are surrounded by a type of cell called a keratinocyte. Late melanomas that are really advanced are surrounded by a totally different cell called an adipocyte, which is actually the focus of my ACS grant. These are fat cells. And so my lab has done a ton of work to try and understand cells like keratinocytes and adipocytes and how exactly they seem to shape the ability of cancer cells. So why do we think this is therapeutically important, right? If you think about the most significant advance that has happened in cancer, not just melanoma, cancer across the board, over the past, 20 years, it's immunotherapy, right? And immunotherapy activating the immune system to fight cancer is by far the most exciting thing that has probably come around. But that is the ultimate example of an inside-out therapy, right? That immunotherapy does not actually target the cancer cell, right? It targets the immune cells that are surrounding the cancer cell. And somewhat ironically, that turns out to be the single best way to actually treat and in some cases cure cancer. So I think there's a real lesson there that targeting the outside of the cancer cell might be more effective than targeting what happens inside the cancer cell. So how do we study that, right? The big problem is it's super complicated to study this, right? Because you can imagine there are probably millions of interactions that could happen. My laboratory a number of years ago decided to study this in this pretty slightly weird way, which is to use these animals called zebrafish to study this. And at the time, that was a slightly weird choice of a way to study this, but there was a really specific reason we decided to. And that has to do with the fact that we have these fish called Casper. Sorry, somebody has their audio. If you wouldn't mind muting, that would be amazing. So we had made these fish called Casper that are relatively transparent. So why fish, right? Why fish? So it turns out that fish are vertebrates, right? So, they're animals. They get cancer, but they have this really unique property that the animals are transparent. And what does that let us do? Well, what that lets us do is to figure out exactly how this inside-outside interaction happens in real time. So let me show you some examples of this. So we can take our fish and we
Sloan
can
Kettering trying to make his study.
I'm sorry, we're still getting some background noise. If everybody could mute, would be great.
So how do we do it? So we can take these fish, we can use genetic engineering techniques so that they develop very naturally melanoma, in this case, using the BRAF gene I talked about. And then using these fish, we can actually see exactly how melanomas start, right? So 3 weeks, after they obtain this mutation, you can see this little black patch here. 5 weeks, it gets a little bigger. 9 weeks, it gets even bigger. And by 16 weeks, it's this really large melanoma. And we can see this just by looking at the fish. And of course, we can label things with fluorescence. So right away, we can see that we have unprecedented ability to actually image the very, very beginnings of cancer and how those very, very first cancer cells start. And this is where we started. We're like, well, this is really powerful because if we could understand exactly how it all gets started and then how it progresses, maybe we could try to interrupt that process. We've taken this a lot further because now we can do things where we can image the fish, like on the left, where we can actually see, where not only does the cancer start, you can sort of see the large green thing in the middle of the fish, but then how it metastasizes. And so you can sort of see here, if you look sort of towards the front of the fish, now you have green cells appearing in new places they weren't at the beginning of it, right? So that's a classic example of metastasis. And then if you look at the video on the right, that's an example of this inside-outside behavior. So in this video, what you see is an individual cancer cell, the round cell right in the middle, interacting with the blood vessels of the fish. Those are the surrounding cells. Those are the outside cells. And so in this one animal, we can actually see virtually every single step that leads from cancer starting to eventually metastasizing and then interacting with its neighbors, right? In this case, blood vessels, but we can do this for lots of other cells as well. And so I call this watching melanoma as it happens, right? Because it really is this unprecedented view into how did cancer start and then progress. Okay, so that's all the slides I wanted to show because I think slides can be actually kind of a little bit, overwhelming sometimes, and I'm going to stop sharing. And instead, what I thought I would do is describe a little bit how we are thinking about using all of this information. So the lab, I would say, kind of thinks about this in two ways, right? One is that we can use this unprecedented level of detail to try and understand what are the molecules that mediate this inside-outside behavior. And so we've had a number of papers that have identified those molecules. And we have focused initially, as I said, on the interaction of melanoma cells with keratinocytes and identified many of those interactions. And more recently, then we've identified the interaction between melanoma cells and adipocytes. And I want to talk about the adipocytes for 1 second, because I think they're unusually interesting. So adipocytes are fat cells, right? Those are the cells that every single human and animal on Earth has. And those are the energy storing cells, right? So every person has adipocytes because that's how our bodies through evolution store energy. If you don't have adipocytes, you can't store any energy. So when you went for a walk or you went for a run, you wouldn't be able to do it, right? Because you need the fat stored in those adipocytes. And based on the images I showed you, we sort of had this hypothesis that the cancer cells appear to be completely surrounded by those adipocytes. And so we had this idea that maybe one of the things that cancer cells do, excuse me, is able to take advantage of this very evolutionary thing, which is storage of energy. And they could actually sort of suck out the energy from those adipocytes and use it to fuel their own growth. And that turns out to be exactly the case. So the melanoma cell interacts with these fat cells, or also called adipocytes. The melanoma cells send out a signal to those adipocytes that say, we want to extract energy from you. The adipocyte responds to that and then sends out its fat molecules. And then the melanoma cell, quite remarkably, then takes up that fat and uses it for energy. And that allows the cell to grow and invade and progress. And we showed in a previous paper that if we block that process from happening, right, we block that communication between the melanoma cell and this energetic adipocyte, we could actually stop tumor growth in that way. And so this is a theme that we continue to be very interested in, right? This idea that interrupting this energetic exchange between cancer cells and their neighboring cells could actually be utilized for therapeutic possibilities. And so we have a couple of follow-up studies now that have looked at this more squarely. One involves this question of how does the cancer cell take advantage of this energy, right? Because that was a mystery in our early studies. All we knew is that the cancer cells sucked up the energy, but we actually didn't have a very good insight into how they actually utilize that energy and what do they do with it exactly. And so ACS basically funded us to figure that out, right? To figure out how that works. And now we have two follow-up studies. One is about to be submitted for publication and the other probably in the next 2 weeks or so that describe in a lot of detail exactly how that works and how does the cancer cell use this energy to help it grow. And that what I think is really interesting about this is that it also opens up really cool possibilities for therapeutic targeting, right? Because a lot of the molecules and genes we've identified in these studies all have the ability to be inhibited, right? So it's sort of a compelling idea to think that perhaps we could come up with drugs that would target this fat energy utilization and try and block that. I'm not saying that's easy to do, right? Finding drugs against cancer is really hard, but labs like mine are the ones that we start out by identifying how it works. And then what we tend to do is hand off that idea of drug development to partners who are good at that kind of stuff. My lab is definitely not a drug development lab. We're not good at that. But there are tons of labs that are good at that, right? Who are really say, okay, Rich White developed this model. They identified the gene or the protein. Now we're going to find a drug that targets that. And that's something I think we'd be enthusiastic about in the future. And I think is a real possibility. And for us, I think the take home message of all of this stuff is that immunotherapy has taught us something that you can't only target what happens inside the cancer cell. You have to target what happens outside the cancer cell. And our vision for this in the future is that the real way or the real path forward in cancer is by simultaneously targeting the special things that happen in the cancer cell with the things that happen outside the cancer cell, like sucking energy out of an adipocyte. And I think there are going to be multitude of examples of this ability of finding drugs that block this inside-outside behavior. And so that's, really a lot of what we do. Obviously, I'm happy to go into a lot of details, but I think that in a nutshell kind of describes the way we think about the problem and what what ACS has allowed us to achieve. So maybe I'll pause for a second and stop. And I'm, of course, happy to take questions. And there's really any question is totally fine with me. So.
Well, thank you so much, Dr. White. This is truly fascinating, and I'm learning so much about all that you do. And you'll see that Sheila actually popped in a little bit of a link so you can look in more detail about Dr. White's lab. And Connie's here saying how interesting it is. So I agree that maybe we can pause a little bit, see if anybody has any questions now that we have learned a little bit more. Robin is asking, does this work apply to all types of cancer?
Yeah, I think that's a great question. I would argue yes. I think this inside-outside behavior 100% applies to all cancers. I don't think there's any cancer that this does not apply. To greater or lesser extents, the influence of, say, the adipocytes will apply to some cancers probably more than others. So, for example, there's very, very good data that adipocytes play important roles in breast cancer, ovarian cancer, melanoma, pancreatic cancer, prostate cancer. But it may not be as important in, let's say, I'm just making up, brain cancer. I don't know. I think it's very hard to say everything applies in every situation, but the general principle of inside-outside behavior applies across the board.
Thank you for that. Rufus is asking, what about solid tumors? Prostate.
Yeah, so I kind of addressed this a little bit, actually. I think I would argue that the inside-outside idea, and especially the adipocytes, are probably most relevant to solid tumors rather than liquid tumors like leukemia. Leukemias have a unique problem, right? They float around the body. Whereas if you think about most solid tumors, they're kind of enmeshed in tissues. And, for instance, prostate cancer cells are surrounded also by fat, right? And so I think that everything I said applies probably most to solid tumors.
Great. Any other questions for folks on the line? We have some great volunteers from really all across the Northeast joining us here today, and it really is so inspiring to hear, your work being done in your lab. Anybody else want to come off mute or ask any more questions?
Anna, I would happily ask a question. I'm curious about the actual structure of your lab team. Like, how many folks do you work with, and kind of what is your day-to-day? Is it Monday through Friday? Like, how does that work for you?
Yeah, it's a great question. I'll speak in the post-COVID mode. I'll give you a snapshot of our normal operation. So my lab is 12 people. It's about half PhD students and half postdoctoral fellows, meaning people who've already gotten their PhD and are doing advanced training. So I usually keep the lab split about that. I would say most people, organize their day-to-day based on experiments, right? And so one big difference, I think, in the research world, let's say, versus medicine, right? In medicine, it tends to be more like a 9-to-5 structure. You come in, you see patients, you go home. Research is not like that at all. It's entirely dictated by the experiments, and therefore it doesn't fit into a 9-to-5 structure very well. And that has pluses and minuses. The minus is people can become obsessive about it and not, work too much. But the plus is that I think he gives people tremendous freedom to explore. So it's definitely not anywhere close to a 9-to-5 kind of thing. But I've never viewed that as a problem per se. It's just that you have to kind of get used to that kind of structure. So.
Great question. We have something else coming through. Apologies if this was covered. Why are zebrafish the best for modeling this type of study versus mice or another model? Can we expect to see more studies adopting zebrafish in the future?
Yeah, that's an excellent question. I always emphasize that the fish is not the only way to study this. A lot of it has to do with the complexity of, in that slide I showed with all these different things that interact, you need a model that lets you study this at larger scale. Mice, of course, we want to be careful in the way we use mice. And each study, you can actually only use a very small number of mice, whereas fish are tiny, right? Our fish are, extremely small. And so that lets us do larger scale studies that would be impossible to do in any other system. I mean, just quite literally physically impossible to do in any other system. The other reason we use fish is the obvious advantages of having a transparent animal to allow us to see every cell as it's moving around, which obviously cannot be achieved in mice, is an obvious advantage for us to be able to track these cells. So I think it really is this combination of scale of experiments that could never be done in mice or any other system, the complexity that is hard to achieve with cells alone, and then, of course, the transparency that lets us watch it all happen.
Thank you for that.
I have one question.
Hi, Kelsey.
Hi, I apologize if you've already answered this, Dr. White. When did you get the funding from ACS? Like, when did that program begin for you?
It was about I want to say 3 years ago, somewhere in the range of 3 years ago.
Okay.
And it's typical for these types of grants is you get the funding and then it takes you somewhere between 4 to 5 years to actually publish the findings, right? As you know, publication is an extremely slow process. So when I got the funding, we hired 2 people to join the lab. One is a postdoctoral fellow, and one is a graduate student. And the 2 papers that I said we're about to submit are their 2 papers. So
Very cool.
Yeah.
Thank you so much.
Loving the questions coming through. We're all learning so much, Dr. White. When you look to fill your team, what type of backgrounds are you looking for? Cell biologist, chemistry, et cetera, that comes from Maureen.
Yeah, that's a really complicated question. I'm a little bit of an outlier here. So because I think I have a bit more of a background in developmental biology, I actually tend to recruit people who do not have a background in cancer biology. So I think I'm actually quite unusual in this one way. And the reason for that is because I think that, well, there's a multitude of reasons for it, but the reason at its core is that I think you need intellectual diversity. And the only way to achieve that is by having people who come from backgrounds where they don't have preconceived notions. What I think happens in a lot of careers is that you really get fixated on a certain idea because that's what your training has told you and things like that. And the only way to get out of that box is to hire people who know nothing about the field. And that has worked, I think, tremendously well for me. There is a cost to that, though, right? The cost is that it takes those people 1 to 2 years to get up to speed, right? So I think only in an academic environment could you tolerate this type of approach. But just in my experience, it's a huge payoff, right? To have this diversity of opinion. Otherwise, you really get into a weird groupthink where everyone kind of thinks the same way.
Interesting. Thank you for that. You mentioned upcoming publications of this work. Where can people find it? That comes from Rob.
Yeah. So we've had a number of publications tangentially related to this in journals like Science and Nature. The 2 publications that I mentioned that will be coming out, the way publication works now has evolved a lot, actually, which is now we tend to post our papers, excuse me, on something called bioRxiv, which is where the papers get posted before they are peer reviewed. And then they get peer reviewed by journals like Nature and Science and the standard journals, and then come out in those journals. This has been a radical shift in the publication world, actually, is this use of bioRxiv. Where we've seen this the most is with COVID, right? Because almost every COVID paper, as we were learning, got posted prior to peer review in a journal on bioRxiv, actually. So my lab's policy now is every single paper, as soon as it gets submitted to the journal, also gets posted on bioRxiv at the exact same time.
Great. We'll all be on the lookout for that. A couple of questions that are interconnected, both coming as direct messages and in the chat. People acknowledging your hard work and losing people to melanoma specifically. Very uplifting to see what a long way it has come. What's a great way to directly donate specifically for this research, and how reliant is your lab on funding from the American Cancer Society specifically?
Yeah, these are both good questions. Let me kind of address both at the same time. So in terms of, how can people directly donate? I think there's a few ways that happens. In some cases, people who want to donate will just literally contact me. And Sloan Kettering has a mechanism, as do all institutions, for direct donations to labs, right? And that has absolutely happens, right? Where people, just call me out of the blue or email me typically and say, I would like to support this, and they make a philanthropic gift, and then we organize it that way. In other cases, it is made to Sloan Kettering with, sort of the idea of being like, I want to donate in general. But of course, you can earmark that for certain types of research. That's, of course, we have an office that does that kind of thing. And I think that both happen, right? I think both individual donations, but also institutional donations. And then finally, your question about how does funding like ACS fit into it? It's quite substantial, but not certainly the only piece of it. All labs that I know of, at least, operate through a combination of NIH funds, right? That's where we get our standard grants from. Foundations like ACS, Melanoma Research Alliance, Mark Foundation, Pershing, that's a whole basket of donation funding. And then finally, exactly what you raised, which is philanthropy, right? Somebody just donates money to a lab. And so I think of those as the three pillars of funding, right? It's federal funding, foundation funding, and philanthropic funding. And every lab I know relies on some combination of those three things.
Fantastic. Learning so much. This is so interesting. I have a question here from Connie. What is the best way to support the work that you do as your audience?
Yeah, I mean, I think that can come in really two flavors, right? One, obviously, this philanthropic support, right? Financial support, as we just talked about. And then the other, and I don't trivialize this, is what might be called advertisement of the work, right? Twitter, Facebook, those kinds of things. And, 10 years ago, we might have been sort of dismissive of that type of support of the work, but now, of course, we're not, right? I think, people have to be aware of what's going on. There's a lot of scientific misinformation out there. And so actually communicating science by people like you is actually quite important. In addition to money, there's the whole other piece of communication.
Fantastic. I have a question. What have made you interested? How long have you been interested? Is it something that you always wanted to do growing up into science and medicine, or is there something specifically that made you want to go into this field?
Yeah, I think I'm one of those weird kids who always kind of knew what I wanted. The joke in my family is that, I had pretty much plotted my entire career path by the time I was 14. And it's actually kind of true, actually. Like I knew pretty well what I wanted to do. So when I was in high school, I started doing research in an HIV laboratory because at that time HIV was a big thing. And then when I got to, my MD PhD program, I kind of oriented everything so that way I could have this type of career. And I think I always knew that although I love medicine and I love seeing patients, that I would probably be more on the research side rather than on direct, 100% seeing patients kind of thing. I think I knew that fairly early on. But, my daughter who's 11 often asked me, she's like, but don't you want to see patients? And of course the answer is yes, right? I love seeing patients, but there are truly only 24 hours in the day. And so those are hard decisions that I had to make. So I would say pretty early, I think I was one of those kids who kind of just knew what I wanted.
Awesome. Your passion is very evident. So it made me wonder how long you've had this passion for
I'd say
projects.
14 is the first time I stepped into a laboratory. So
fantastic. Sheila is asking, what are you most excited about or hopeful for with your research in the next year or two? Great question.
Yeah, that's a really good question. I think that we are really, really excited about this, really understanding at a deep mechanistic level, this idea of inside-outside behavior, right? How do cells communicate in exceptionally complicated ways? And then by identifying that in all of its complexity, right? How do we begin to turn that into therapeutic opportunity? Now, as I said, my lab is not a drug development lab. That's not what we do. But almost everything we do has therapeutic implications, right? And so I get really excited about this idea that we can target cancer by going after this inside-outside behavior, which up until fairly recently was not a big idea in cancer, right? It was sort of ignored for a long time. But I don't know, I've just always had this faith that that will ultimately be the way to target cancer. It's kind of like if your only goal is to kill cancer cells, they're going to resist that, right? And so I think you need to be more creative than just saying, well, we're going to kill every last cancer cell. I don't think that's going to work, right? Because they're always going to find a way to resist that. So I think the more rational way to do it is, how do you target this inside-outside behavior to control it, rather than just say, I'm just going to kill every last cancer cell? Because I think that might be hard to do.
Interesting.
And that's what immunotherapy does, right? That's
Mhm.
exactly what immunotherapy does, is it lets your body almost like just deal with it.
Mhm. Fascinating. To add to Connie's question, how can corporate partners support you? Is there a need for in-kind of any kind or boost grants?
Yeah, I mean, that has definitely happened. In fact, that has, happened on a few occasions. There's a few ways that happens. The NIH is a little funny with in-kind grants or boost grants, actually, but foundations are not generally, right? So like, let's say, I have a foundation grant from ACS or another foundation. Corporations are encouraged to provide those type of in-kind grants. And in some cases, corporations, if they have a philanthropic arm, like Google, for instance, has a philanthropic arm, will directly support labs, right? And they'll just say, we think this research is important, and we'll either partner with a foundation to give an in-kind grant, or in some cases, the corporation's development arm will just say, we're going to donate X number of dollars to this laboratory, or to support this type of work. So that absolutely happens, and it has happened to me. So.
Great. Such great questions coming through the chat. And we're having such good conversation. I'm loving it. Maureen.
And I will say, any lab that says, we don't need that is crazy, right? We all need support because doing research is just crazy expensive,
Oh, yeah.
So.
Maureen's asking, are there other labs looking at cancer from the inside outside behavior? And is that becoming the norm?
I wouldn't call it the norm. And I think there's both historic reasons for that, because most labs have not approached it that way. So I wouldn't call it the norm. I would say it's accelerating dramatically, right? This idea, because the tools have gotten better, right? So I think, science, there's always this chicken and egg question, right? What comes first? Does the technology drive the science, or does the science drive the technology? It's like this classic chicken and egg question. But in this case, I think it is that the technology has helped driving the science, right? We've gotten better at analyzing inside-outside behaviors through single cell sequencing and things like that. And that has propelled people to now think about this for their own laboratories. So I think of this as a dramatic acceleration in this field.
Fantastic. Well, it looks like our chat is quieting down, at least in the chat box. Wanted to be sure we opened things up for any other questions. I'm going to pause right there before we take any more, just to, again, thank you, Dr. White, for this fantastic overview of the work that you're doing in a lab. It really is such a privilege to be able to spend this time with you one-on-one, and you really just make it so easy for us to understand, those of us who aren't scientists. You really just make us also comfortable in asking these questions and learning more about the work that you do. So thank you from the bottom of our hearts, as American Cancer Society staff and volunteers. This really is a privilege for us. Anything else from folks in the chat? I know we've had a lot of great conversation before, we wrap things up here.
Anna, I just want to jump in too, and thank you, Dr. White, for your time. This is like by far the best lunch break I've ever had in my life. I really love hearing your insight, and I love that you think outside the box in many ways. And it's just very cool to hear the specifics about what's going on in a lab, when sometimes day to day we hear a lot about research out in the world and all this big change that we're pushing for, but to see it kind of happening not only from a lab, but a lab in our state is really cool. So thank you for taking the time to talk to us today.
Yeah, no, I appreciate that comment. I think it's a really important point, because most people don't actually have any experience in what actually happens in a research laboratory, right? And so I think that, it can be really and again, with the theme of science communication, I think sometimes people think that mysterious things happen in labs, and it can seem mysterious, but it's not, right? There are nuts and bolts things happen, and the more we communicate that, the better it is for the country, for the world.
Fantastic. Well, it does look like things are quieting down in the chat. We did have some great questions come through. Sheila did pop in the chat a direct link to Dr. White's lab if you want to take a look at that or reach out to him. But again, we're so thrilled to have you as a partner. Can't thank you enough for spending some time here with us today and being flexible with us, as there was no Wi-Fi in the actual lab.
Sorry about that.
Did you just come up with those slides in like the 3 minutes before?
Well, I kind of, took various talks and,
I
so
mean, that was incredible. We were able
I give a lot of talks. So
we were able to, work through that in just a few seconds.
Yeah.
Before we go though, we do have one more question from Christy. Do you actively collaborate with other researchers while working on your own published materials, or what does collaboration look like for you?
Yeah, that's an excellent question. And, I might have given you an impression that we kind of do this, as an entity to ourselves, but nothing could be further from the truth, actually, right? So we collaborate with people, massively collaborate with people at Sloan Kettering, outside of Sloan Kettering, NYU, Oxford, Harvard. I cannot fathom a research laboratory being able to do this today on their own. It's just, science is too diverse, it's too complex. So we collaborate with stem cell biologists and with mouse people, drug development people. We have a paper coming out very soon that has, I think, 40 collaborators that include people from Sloan Kettering, Cornell, NYU, Oxford University, Cambridge University, and several universities in Australia. Right. And I think that, to me, is the essence of research, right? Where we cross over these world boundaries, and we couldn't have done it without these other laboratories.
Great. What a good way to wrap things up. Thanks for taking that last question. Thanks for being with us here today, and keep up the fantastic work. Thanks for all you do.
Well, thank you, everybody. And I hope everyone has a good afternoon. And feel free to email me directly with questions. My email is widely available all over the web, and don't feel shy about emailing me. So
great.
All right.
Well, thanks so much. Have a wonderful day, everyone.
All right.
Thanks
Hope
for
everyone
joining.
has a good day. See ya. Bye-bye.